drug discovery, drug development, working with the FDA, getting drugs approved. So that's the sort of optics I bring to how I look at each of these clinical trials. So I try and take that and guide you through what's going on out there so as you can walk out of here this weekend a lot smarter. If you're not, that's my fault. I didn't explain it well. So you hear this strange accent. So stop me, if I tend to talk too fast, or stop me if I don't explain something too well. Because I view this very much as a meeting for you. And there's why I want to make sure that you walk out of here really knowing what the landscape's like and how you make judgments about clinical trials for your son. It was also a great segue, to hear the TRINS team, talk about what a clinical trial is and what a clinical trial isn't. These are experimental medicines. The fact that you're in a clinical trial doesn't mean to say that that drug is work or that drug is safe. That's why we do clinical trials. So it's an important aspect, to get into your mind that you select the right types of trials. You're most informed about how you make decisions into that. And that's an important part of this here as well. Another thing really to sort of think about is the context of where your son is on the natural history of this disease, because not all trials are applicable to every individual. You'll see some of them are very applicable to very young boys. And they're on their sort of stable growing phase. Some of the trials are they're looking at different parts of the mechanisms of this disease. And you'll see that there's for older boys, particularly boys that are non-ambulant. And so those trials are really specialized. Like, for example, an antifibrotic is really pertinent in there, or drugs that are targeting cardiac are for older boys, because there's whenever it manifests. But a lot of the others are for younger boys. And really, we're understanding where you are in the natural history is really an important part also of trying to sort of figure not just about the mutation, but what trial should I be on, and how's it going to impact my son, and how's it going to be really beneficial. So there's a lot of complexity, and I know there's a lot of complexity in there. And so what I want to make sure is that I can explain it well to you. So don't hesitate to stop me or saying, hey, you didn't explain that, or I didn't understand that. And then after this, I'll be around for a while. I'm here until tomorrow afternoon, so there's tons of time if people want to sit down and just talk in private, and you might not want to ask questions publicly. People always feel, I can't pronounce what the heck that there is, so I'm not going to ask the question. Anything will do, so I answer most questions. Let me give you a sense of the vision that we looked at this, because I've been doing this for quite a while, with Deborah and the team, as I mentioned. And whenever we first started out on this, there was only a couple of companies that were involved in clinical trials in Duchenne. And they're still with us today. Those companies are still here and still focused. And you can see the development of their pipeline. But today, almost every major pharma and biotech company is active and interested in this space. And there's because there's a tremendous amount that we know now about a number of things. Part of it is we know much more about the science of this disease. And you'll see also the fact that the latest cutting edges of technology are really being thrown at this. So you can see like genetic approaches into this that really weren't there 4 and 5 years back. And like all of that there collectively on top of, there's real granularity today about understanding the natural history of this disease. Whenever we first walked into this trying to do clinical trials a decade ago, we didn't really understand all the nuanced natural history. And you have to understand that in order to really know what you're going to look as a clinical endpoint. And we've got a great handle on that there today. That there's really evolved over the last 3, 4, 5 years. And it was touched upon with all the different databases that have been worked on where we've been following boys for many years. So the disease is absolutely in the right place right now in order to be conquered. And I can see that with all the different science that's going on out there, all the different companies. These companies, for the most part, are not limited either by human manpower or by financial capital. So we're in a really strong position, I think, over the next few years to see some very significant changes. And so I'll talk through some of that there. I'll share something with you, because I was thinking about it earlier on as Tiffany was talking about a mother's journey into this and giving you a sense of what a scientist's journey is. And I can tell you, I'll speak for me, but I know that I'll speak for every one of the pharma companies that are out there. There is the science in order to defeat this disease is there, and it's a matter of time working our way through all of this. And the vision that we have really is over the next handful of years that the first step in the journey to get to where we want to be is going to be a combination of therapies that will work together in order to reduce this disease into a chronically treated condition. And by that I mean drugs that can actually increase the amount of dystrophin, if that's possible in a boy, or drugs that will actually be new mechanisms to fend off the inflammation, these new anti-inflammatory drugs that you'll see, drugs that will work in order to regenerate new muscle in the boys, are drugs that can actually target as an antifibrotic agent. And so whenever I walk you through the pipeline and you'll see what's going on out there, I want you to frame it in your mind of there seems to be a lot of stuff out there, and the purpose is really over the next 3, 4, 5 years as some of these drugs are approved, that a combination of them together, targeting different parts of the mechanism of this disease, will actually reduce this into a disease that can be chronically treated rather than being a burden or a life sentence on somebody. And that's where the first step of this is. Towards the end, because I'll focus almost exclusively on stuff that's in the clinic rather than stuff that's in preclinical development, but we'll touch upon some of the work where you could actually see a cure, where you've got an ability to go back and correct some of the DNA faults that are in there, and that's possible and practical and feasible. It's not close to the clinic right now, but I'll share that with you because that's where we see the 5 to 10 years out. But really what we're going to talk about now is things that are in the clinical trials that are working their way through to approval. And they may seem to the untrained eye, snapshots all over the place, but put it into your mind of how these are going to work together, because there's really what these different companies are working towards. And there's an enormous amount of collaboration that goes on with individual companies as they share data and share approaches. So I put this up. I've got a couple of slides that really just talk about the disease to begin with, so we've got a level playing field of what we understand. And this is a cartoon of the exon skipping map, or it's the reading frame for dystrophin. And this reading frame is a little bit longer. Dystrophin is probably one of the largest proteins that we have in our body. And that's a challenge in a number of ways. It's a challenge to the technology of trying to restore it, but it also speaks to the complexity of what dystrophin actually does. It's not a very simple protein. It's a very, very complex protein with lots of different function. And whenever it's lost, it probably speaks to why this is such a devastating disease. There's a tremendous amount of roles that dystrophin has. Very large protein, and it's made up in muscle cells with these individual blocks, 1 through 79. They call it the reading frame. It's made inside the cell. Number 1 will join to number 2. It's not made all at once. It's made in these individual blocks. And these are all spliced together inside the cell. And that there gives you the full-length protein. So the way that nature's designed it, on the beginning of number 1, there's a sign that the machinery that's inside a cell will say, start here. And it'll start to make the first bit of the protein. And then it'll splice number 1 to number 2. Splice number 2 to number 3, all the way through to 79, where there's another sign that says, stop, you've finished. And that nature actually uses those sorts of codes within inside the cell, stop and start, and we'll get on to that in a second. The complexity of this is sort of, it's hidden in the cartoon. I won't get into any more than that, but some of these blocks are rectangular. Some of them have got funny edges in there. And I can explain that to people, and it's just not really worth digging into the science of why that's like that. But that's the way it is. Some of these blocks will join on only to their partners, and other ones are quite uniform. And you can see the nature here of what happens whenever something's missing. I've given an example where exon 50 is missing, and I picked that because it's probably the most common exon that we find deleted within the reading frame of boys. And there you'll see that 49 and 51 won't join together. They're not, as the way the cartoon is drawn it, they don't have the same sort of edges. And the only way that this would work is that 51 is removed such that 52 and 49 would join together. And that's a good example. So if you remove 51, then that's what's going on with exon skipping. We can splice out 51 so that it's not removed, it's just hidden. And then the splicing machinery reads over the top of it and then joins 52 onto 49. And there's the whole basis behind exon skipping. And a couple of things. One is, for these, this is obviously a very personal type of treatment for an individual who has that very specific exon missing. If you've got another exon missing, like 52 or 33 or 12, this is no good for you. There's why known mutations is a really important part of this. So to explain this in terms of what nature has taught us, this is not something that we've invented looking at this as a series of building blocks coming together. We're following what nature has actually taught us. So there's a database that we have that follows Becker muscular dystrophy patients. And the Becker patient is one where you've got an in-frame truncation of the dystrophin reading frame. And this is a good example of what a Becker mutation would look like. So there's a significant number of Becker muscular dystrophy patients that are pretty much asymptomatic for this disease, that have exactly that mutation, 48, 49, 52. So as we're trying to do this with exon skipping, we're not following building block rules. We're actually following what nature has actually taught us, and that there's the template that we're using in order to do that. And I can give you lots of examples of this, but it speaks to why exon skipping is a very important part of the pipeline that's out there, because we're really mimicking what nature has done already. So another example of this is to show that truncations can be functionally important. You can lose exon 45 all the way through to exon 55. That 10 exon block can be excised because we find that in Becker patients that have got a very mild, very asymptomatic disease. So that whole 10 exon piece in the middle gives you a shortened form of dystrophin, but it's a very functional form of dystrophin. So that gives you a sense, that, yeah, nature doesn't need it all. It's designed it for reasons, but there's certain mutations in there that it's actually taught us that we can start to mimic, and that there's the basis behind why we do exon skipping and why it's important for us. And I'll get on to some more about this in a minute, but I think that it speaks volumes about what we can actually do. That whole exon block between 45 and 55 is known as the hotspot. The vast majority of deletions and whatever really occur within that there region. It's just that part of the gene that seems to be very prone to mutations, and that there's why we've honed in on that and recognized that you can actually remove large chunks of this protein in the right way. It has to be in the right way, and not just all of it, but in very specific ways, and nature has got there one step ahead of us. So any questions about that? I must be explaining that really well. Okay. I put this up to remind me of a couple of things just to talk to you so you can get a sense of it yet again. And this is the full normal reading frame where I told you the translation starts. You get the start sign, you get the stop sign. But I put a few of these in just to, again, highlight the points I've made on the last one and what you actually see whenever exon blocks are removed. This here is a fairly common one where you can see 47 through 50 is missing. And nature tries to read through this, but what I want to try and tell you is that it can read through, but it produces nothing but junk protein. So as it goes down each one of the codons, every one of these little asterisks is an intermediate stop sign that's built along there. So the protein is there, but it is jumbled protein. And that's what happens whenever you lose the reading frame. Another example here, and it speaks to the approach that PTC is doing, is not where you've lost an exon, but you've simply lost a single codon within the sequence. And that there can change the sequence from an amino acid into a stop codon. And that's what we call a nonsense mutation. Roughly about 13% of boys have got exactly that there. And this just shows a nonsense mutation, is this right there. Translarna, that's in trials here, and we'll talk about it in a little while, in the United States and conditionally approved in Europe. It is designed to specifically read or force through these nonsense mutation stop codons and actually produce full-length protein. And that there's the basis behind that. Very different from exon skipping, and it's addressing a very different specific issue that's out there. That's what I talked about in the last one. You can see a NIN frame. This is 46 through 54 deletion. And there's a classical Becker mutation. And again, the Becker database that we have is loaded with a lot of examples of individuals like that, where that is a very asymptomatic, very mild disease. So you can lose those exon blocks, as I said before. Duplications of the gene can be quite traumatic, and you can see the example here. This is one that Deborah touched upon actually right at the beginning. This is the most common exon that's duplicated in humans, exon number 2. And when you've got 2 copies of number 2, you can see the reading frame is perturbed and nothing but junk protein and stop codons that are in there. The difference here between deleting exons is that if you can remove one of those 2 copies, you get full-length normal dystrophin again. And that there's been the basis of the work that we funded with Kevin Flanagan, and that there actually has been done very successfully. We built a mouse with a duplicated exon 2 and was able to skip and remove just one of them and restore the normal protein. And that their product opportunity, as mentioned earlier, should be in the clinic in the first part of next year. the reason I point this out is that it's not only technically feasible and one can do it. Whenever companies are looking at exon skipping, they're going after the larger patient populations. You know, there's 13 or 14, 12 to 13% of the boys are amenable to exon 51 skipping drugs, but there's less than 1%, much less than 1% of the population that have duplicated exon 2. So we figured that trying to start at that end and supporting research at that end, you know, gets rid of that mutational envy. You know, and so somewhere in the middle, what the companies are doing on the large exons and what the academic groups are doing on the small ones will meet, and a lot of patients who are amenable to exon skipping will get that sort of product. The last one I put on was just to remind me, you know, how much you can actually remove from this. There's a paper in the journal Nature that came out quite a number of years ago, and I constantly remind myself of it because the reason I put this on, I'm going to talk about it again when we talk about the mini gene work. This particular gene is one where you've got an in-frame deletion from exon 17 all the way through to exon 48. That's more than 40% of the gene that's actually missing. And that's an enormous amount of the protein for something that is so long. And you'd look at that and go, that's not functional. The paper in Nature described a family who presented into the clinic with typical myalgia. You know, this guy was walking with a cane at 60-odd years of age. He had a number of family members that were exactly the same. And you'll recognize the story. As he went in, they looked at CK levels that were elevated and immediately went, oh, we need to look at you much further. And as they looked at muscle, they found that he'd lost over 40% of the dystrophin gene. And the importance of this can't be lost on us. You know, you're looking at that as a significant loss of that their whole protein is still associated with a very mild phenotype. They had a normal life. He was walking with a cane at 60, which I would allow anybody to do. You know, I would take that. And I think that there really speaks to, we can go quite short with this. We don't know how short. That there's I would say that's the most that we've seen removed from the gene and still have it functional. It doesn't mean to say that it's out there, just we haven't found people. They've never presented. We don't go looking for this sort of stuff. But you can remove an extended amount of protein from this and still have a functional dystrophin gene. And that becomes an important component in the gene therapy discussions that we're going to have in a little while and later on this evening. Any questions? You guys are real quiet. Go on.
I have a question.
Sure.
So, do we consider an in-frame, so in our instance, we have an in-frame duplication from 13 to 44.
Mm-hmm.
And they thought he was sort of an outlier at first. And they said, okay, if he stops walking at 12, we'll consider that Duchenne.
Mm-hmm.
So I guess I'm just curious, when you were talking about Becker, you were saying in-frame. Is in-frame typically considered?
Yeah, so there's a couple of things to point out, and this sort of, I'm going to stop myself from getting into the weeds of the science, and if I do, hit me a kick, okay? But hidden behind all these building blocks is the complexity of protein structure and function, and it is hideously complex. You know, there's secondary structure that has to join together in order to give you something that's functional. Building blocks don't tell you about that. It gives you the very first, hey, is there a phenotype-genotype sort of relationship? Hey, if the building blocks work, then you may be okay. But not all of them follow that, and they don't follow it simply because the tertiary structure of the protein really demands much more than just simple building blocks added onto it. And in your son's case, you know, that there is a significant duplication. One would have to actually look and see exactly where on the protein that's happening and how, and I won't get technical, but how all of that there stuff lines up in real life. Right. And that there's really what's hidden in there. And no one speaks about it until you actually ask in a very specific question about one point on the protein. And it is that complex. Like I can tell you that you can get to within full-length dystrophin and have only one single amino acid change out of the tens of thousands that are in there and have a protein that is not functional at all. Because you can hit conserved residue, you know, that really changes. It's important for function, important for various other things. So it's not as simple as I'm trying to make it to you. I want you to come out with a good understanding, but there's not going to be a test after this to make sure you've got everything about it. Did that help?
Yes.
Okay.
Thank you.
Any other questions, guys? Sure.
Is that exon skipping will help any mutation? I mean to say like a duplication, deletion, nonsense, everything will work out, or it's specific to only one exon skipping?
No, so exon skipping will, first of all, it is a very specific specifically designed oligonucleotide only for one region of the protein. So something that's made for the skip exon 51 will not affect or will not help or will not skip any other exon on there. They have to be redesigned for every one of the positions. There's a majority, in fact, I think it's on the next slide. Let me get to the next slide and I'll actually answer that there, because there is not all mutations and not all, depending upon where you are in the protein, is amenable to exon skipping at all. And that actually speaks to what happens to those boys. There's about 40% or more of boys for whom exon skipping will have no help. And so what do you do? And that's why we have many other proteins out there, many other approaches, that are not mutation specific. So I'll dig onto that in a sec. Okay, I think you got a sense of why, like a little bit about the science and why we think exon skipping is important, and why, based on this sort of information, many gene approaches are starting to become much more interesting and probably doable for us. To your question, you can run around this pie chart and start to understand where all the mutations lie and the percentage of boys that'll fall in there, but there's a significant chunk for which exon skipping won't work. And where it typically is in the front end of the gene, where it becomes really important at tying it down with inside the cell, and at the back end of the gene, the later numbers, the higher numbers, where it becomes really important in a strange way, for binding proteins and tying it to the cell membrane, they really can't be skipped. There's not good examples of that happening successfully. So for boys with low numbers and high numbers, you tend to think of much other approaches rather than just exon skipping. And we tend to look at it just in a general way. There's about 2/3 of boys for whom exon skipping could be beneficial. Okay? So in answering the question, I've talked to that slide, so it really gets on to, let's see what's going on out there. I've talked about this briefly, but there's a number of ways that we're leveraging this information based on the understanding of the gene and how we're being taught by nature of what we see in Becker individuals. And each of them are represented on this. There is the RNA-based approaches of exon skipping, nonsense-mediated read-through, DNA-based. This is a DNA disease, and we're approaching it trying to fix the RNA, which is why you need to inject every week or every month, because you're turning RNA over. But if you can go one step upstream and actually correct the DNA, then that's possible. And in certain cases, we've been able to do that there with CRISPR-Cas9. And we may talk about it later. CRISPR-Cas is not something that we've invented recently. Nature got way ahead of us a few billion years ago, and it's been around. We've been able to take it and reprogram the gene to actually find very specific parts of DNA that we wanted to cut and re-edit, and it does it with great precision. We can go inside a cell that contains a few billion letters of code and find the one that's wrong and cut it and fix it. The issue with this is one of absolute fidelity, to make sure that you're doing it with 100% certainty, not 99% or 99.9. You want to be absolutely sure that what we're doing in there with cutting and editing DNA is 100% right. And so we know the science works. We know that there's going to go forward into human development, into clinical trials. But for us right now, we're walking through this very carefully to convince ourselves and the FDA with all the safety information that's required in order to get that into a clinical trial setting. So I would say we're a handful of years away from seeing CRISPR-Cas9, probably 4 or 5 years in a Duchenne setting, because of the demands really placed on muscle and how much we have. But it is something that's on the horizon. I'll also talk a little bit about gene replacement. There's a number of approaches in here, from utrophin upregulation, many dystrophins that we'll talk to, because I think that does become one of the most interesting and certainly talked about and closely followed projects that are in the Duchenne space right now. And the other that I mentioned really is it starts to bring together that table that you can see. Hey, this isn't just companies going crazy. There is a real plan behind this and why we want all of this to work together. The muscle regeneration programs, all of them in the clinic, antifibrotics, mitochondrial, epigenetics, the cardiac-focused ones of Capricor, as well as new anti-inflammatory agents, and some miscellaneous ones that I've got on that. And I didn't put it miscellaneous really as anything more than they have multiple functions. Whenever I look at something like the gavinostat program, for example, that there has the ability to work on a couple of different aspects of Duchenne, not one simple single one. So digging into a few of the programs, just to talk about them. Everybody knows and has followed what's going on with the eteplirsen and exon 51. I think some of the company reps are going to be around today, so you may get a chance to have a talk to them. There's not much that I need to say here, other than the thing that's really hot in this area that parents are following is that we recognize some of the limitations of these exon skipping drugs. They haven't fulfilled the hope that we had in the non-clinical studies. In the mouse preclinical models, these were much more efficient than what we've seen in the clinical setting. And the amount of dystrophin that's being restored in here is fairly low. It's in the order of about 1% or 2%. And that's not sufficient for us. We really need to do so much better. And the company knows that. So for the last many years, they've been trying to find a way in order to increase the amount of exon skipping component, these oligonucleotides that can get into the nucleus of the cell. And they've successfully done that there at last. The challenge is that these are very, very large molecules that are hard to cross membranes and get to where you want it to be. And they're also not charged molecules. So in order to get around this, they've got in the clinic right now what they call the PPMO. The first P is important. That stands for peptide modified. They've put on a cell-penetrating peptide, and it's literally just put onto the front of EXONDYS 51, and that allows these to get inside not just the cell, but also the nucleus of the cell in very high concentrations. It sounds really simple, but the challenge that the company's had over the last many years is it's really limited the speed with which they can get them into the clinic. And so after about probably 4 or 5 years of work, they've now got their first PPMO in a phase 1 clinical trial right now. And we'll learn before the end of this year's out essentially what that's looking like. That trial is just looking at the safety. And this is something that Lauren and team talked about, the safety and the pharmacokinetics. And they're going through single ascending doses of it. Now, the reason I wanted to take a minute to talk about it was that the non-clinical profile of the PPMOs is very different from the unmodified drug. The unmodified drug gives you limited amount of transduction, very little really in the diaphragm, very little in the heart, and some in skeletal muscle. But in the PPMO, you're getting massive amounts of exon skipping in all tissues, in the heart, in the diaphragm, smooth muscle as well as skeletal muscle. And so it's a very, very different profile. And if the safety profile of this works and the molecule continues to look well, then we'll go into meaningful clinical trials next year with this, and the company will be focused on seeing how different it is and what the benefits are. But the expectation is that this, if it continues to be safe, should be much, much more efficient than what we've seen with EXONDYS 51. So this is something to watch over the next few months. NS Pharma are another company that's involved in exon skipping. I just wanted to walk through each of the different players that are in here, because the importance of exon skipping really is founded on what nature's taught us as far as Becker muscular dystrophy patients. NS Pharma are working with a compound called NS065. This is targeting exon 53, very different from EXONDYS 51. They've just published their early phase 2 results earlier this year, as well as the World Muscle Meeting. That data looks quite interesting, and right now they're in that space between reading that data and designing what the next trial is going to be. So they're not actively recruiting into a trial right now, but I expect as the company comes through this, we'll learn really how they're going to move this forward into the next round of trials. Wave, a company here on the East Coast, had to think for where I was actually, company here on the East Coast, and they've got an exon skipping platform that's very different from any of the other companies that are out there. They're not using PPMOs, they're using what's called a stereopure form. I struggled to find a little bit of the science to explain that. But this stereopure form of their exon skipping drugs is designed to work at much, much lower concentrations and be able to more efficiently transduce muscle. And they're in clinical trials right now. They're actively recruiting patients. You can see the data that's on your handouts. They expect to enroll 40 patients. Oops, excuse me. Sorry about that. They're expecting to enroll 40 patients, boys 5 through to quite mature. All of that data is on your handouts. And one of the questions that was asked, I think, earlier on from this side of the room was about the clinical trial sites. I've put each one of these as a hyperlink on that. So if you follow that, it'll bring you straight into the details for all those clinical trials that are there.
This just helps you, they don't have the slides at this moment.
Oh, okay.
But we can send them to anybody afterwards. We're just going to print them and copy them.
Okay.
Anybody who wants them, please let us know. We'll send them.
So that's exon skipping. I think that you've got a real sense of that.
On exon skipping, is any of the company thinking or starting other exon skipping? Like we have 7 deletion, we need exon 8 skipping. So like any plan of any company starting? Or
Exon-
like if they start, like how much time will it take? Will it take like another 5 years for the research and all?
Yeah.
So,
Yeah, let me try and dig into that. The question, you know, this you need an exon 8 for your son, an exon 8 skipper. And so I'm not aware of companies that are involved that low down on this yet. A couple of slides back there, I had a bit of a pipeline up for Sarepta. And this is other programs that they've got, 53, 45, et cetera, that they're pursuing. And that there represents larger patient populations, but exon 8 is not one of the ones that's in there. But let me just address the second part of your question. Not really the amount of time that it takes for these to actually go through. EXONDYS 51 started almost 10 years ago in order for it to get approved. This is new cutting-edge technology, you know, really changing the RNA within inside a cell. And it takes that much time for us to really fully understand both the efficacy as well as the risk profile of a molecule like that. Turns out that it's very, very safe, but it does take quite a while. And for the first time in, the FDA will want to see all of that laid down. And that's one of the reasons those trials took quite a long period of time. It's also one of the reasons why the company went into the largest population, because you can find those boys and actually follow them for quite a period of time, rather than, for example, exon 8, where there's only a smaller number of boys. So the information that we've learned there is one where all of that their chemistry and the exon skipping and the risk-benefit profile of the molecule can now be leveraged to the next round of drugs. So there, for example, they're expecting these exon 45 and exon 53 drugs to go in front of the agency within the next handful of months. And they've been in development for only about 4 or 5 years. And all of that knowledge and know-how that has really built up between both the chemistry, the toxicology, the way they're made, the way that you can go into the clinic, all of that's been leveraged in every one of the follow-on programs. So you get that there sort of acceleration effect that goes. And I suspect that because you're going into smaller populations of patients, at that point, the follow-on drugs will be much quicker as far as the approval, unless something unexpectedly happens. But if it follows the same sort of predictable pathway, you know, based on all the information that you've built up, then the FDA, I suspect, and everybody suspects, it's the business model for the company, that all of these follow-on exons will be approved much, much quicker. And when you get down to very low numbers of boys, this actually came up in one of the questions this morning. Whenever you've got a small number of patients and you can't effectively recruit a large phase 2 trial, then in the case where you've only got, I mentioned the duplicated exon 2, where there's only about 100 boys in the country, that there, I suspect, is going to get approved after a phase 1b trial. If you're able to show efficacy, which is probably dystrophin production, as well as measurements for safety and following those boys, you can't do any more meaningful trials after that. And so the FDA would conditionally approve something on that. And that's really our expectation for something like that to happen. Sure.
Apart from one particular skipping of exon, is there any research going on where we can skip the multiple exons?
There is. In fact, there's been so the question is about, can we skip multiple exons? And this has been a goal for a number of researchers for quite a period of time. And part of the driver behind it is, I talked earlier on about, you can skip that 10 exon block from 45 through to 55, and that is a known very mild Becker phenotype. So if you could remove all 10 of those.
Any trials going on?
No, there's not, but I will explain. So that is really the motivation behind that. Now, I mentioned earlier that there's this mutational hotspot on the gene. There's a significant number of boys that fall within that there 10 exon range. And it turns out that if you can have something that skips both of those and removes that 10 exon block, you can cover about 2/3 of all Duchenne boys that are out there. Now, that's been done in mice right now. But the challenge that we've had is EXONDYS 51 skips to about 1 to 2%. If you're going to do it twice, the numbers just fall, and it's not that efficient. And where I think this may become real is if the PPMOs work, or if Wave's stereo pure technology works and exon skipping becomes much more efficient, then we've got a chance of being able to do something like that, and it brings that back onto the block again as something that is a real meaningful approach. And the benefit, like I just repeat it because it is important, if you can remove 2 and you remove that whole 10 block, you can effectively treat almost 2/3 of all boys straight away. And that's the motivation behind that. So it's a good question. Any other questions out there? I must be doing a great job in sort of explaining this stuff.
I have a question on
See, there.
CRISPR-Cas9 also. So the CRISPR will like cut the unwanted exon and will kind of skip it, or will it insert the deleted portion?
No, it won't. So what we've got now, and the approach that's going forward with exonics is a very specific approach. So CRISPR has the ability to do multiple different things. You can go in and do 2 cuts and actually give it a template that you could repair with, or you can go in and do 2 cuts and have nature do its own thing and it'll try and repair the DNA back. The approach that exonics has taken is a single cut. So they've recognized a very unique position that's on the gene around exon 51, and they can use a single cut on both sides and have it a needle to remove exon 51. And that there allows them now to treat a population that's similar to exondus. So the approach that they're moving in is an exon 51 permanent remove or permanent cut. Okay, I'm going to shift gears here. Oh, stop codon read-through. I've already touched upon this. I don't need to say too much. Translarna is a molecule that's conditionally approved in Europe from PTC. That approval was reapproved again for the next while, which is great for European patients. You know, we continue to follow them and learn more. The FDA has turned around to the company, as you're all very much aware, and said we want more data. And that data really has to contain dystrophin data. And that's what the company's doing. They've got a number of trials, the one that's listed on this, and in particular the 041 study, that they believe they're going to collect data and actually show dystrophin levels in there for the agency. And that'll be the point. It's going to take another while for them to complete that trial, get that data in front of them. And that there's really where the FDA are holding their feet to the fire to say, we want to see that data before we actually approve the drug. And they are recruiting into that. Probably one of the most closely watched areas of what we're doing right now is the mini-dystrophin work. And I want to talk through that there because there's things that I think are incredibly exciting in here, and there's a ton of things that we don't know about it, which really tries to sort of give us a level playing field about how to read it. There's 3 companies involved, just to go through them. Sarepta in conjunction with Nationwide, Pfizer, and Solid. All of them actively recruiting right now, you know, given some of the issues that a couple of them have had. There's a ton of stuff that I could say about this that's, I think, really interesting, really exciting, and I think we're right to be excited about what we're seeing in here right now. But, you know, as a scientist, I tend to want to sort of step back and take a deep breath, and I need to see the data, and particularly the longitudinal data. So before we get into it, let's just look and see what each of the companies are doing. This is a microdystrophin, and if you recall the paper in Nature where the guy had lost 40% of the gene in a very, very specific region, there's the shortest we've seen in humans. These are delivered by a virus. It's an adeno-associated virus, an AAV, and that's pretty small. The packaging constraints of a virus dictate that we actually have to remove almost 2/3 of the dystrophin gene in order to get it to fit inside there. And each of these companies have gone and looked at that approach in a very specific way. They've each designed unique forms of microdystrophin that they believe are going to be stable. So there's 3 shots on goal that we've got in here, 3 different forms of microdystrophin, all designed differently from each other. Some have contained binding sites that they think are important for function. Some are cut and joined in regions that they think are going to be important for protein structure and longevity. So there's different ways that the science has really tried to piece these together to fit within the constraints of what you can pack inside the virus. They each are using different promoters, and they each are using different viruses. So if you think about that, if one fails, it doesn't mean to say that another one will, or that all of them will. They've all got very different promoters, viruses, as well as mini-genes. So it is 3 unique shots on goal that I think give us as a community, you know, the confidence that we're going to test this hypothesis and see really what happens. And the questions that we're asking is, We know that we can deliver the mini gene. We've seen it in muscle. And the question right now is, okay, how long does it last for? How functional is it? How do we redose? What is the optimal dose? And I'll explain some of those questions to you. There's a significant percentage of boys, in fact, of us, that have seen AAVs at some point. They are quite common. It's a quite common virus. It doesn't cause infections in humans, but we build up antibodies to them. So roughly about 30% of Duchenne boys that are out there right now, that's a sort of hand-waving number, will have significant neutralizing antibodies, and they won't be applicable for these trials for that reason. If you've got neutralizing antibodies, they'll really attempt to neutralize the virus and the packaging constraints before you can get it inside the nucleus of the muscle cell. So the boys that are going into these trials right now are ones that have very low neutralizing titers, and that there's part of the inclusion criteria, and that makes real sense. You know, as we really go forward with this, there's maybe other things that we have to do to try and reduce the neutralizing antibodies in boys. So the first thing that we're looking at here is the transduction, and that there's been shown, at least Jerry Mendel has shown very successfully, most recently at the World Muscle Meeting, I think in the fourth boy, where there's very high levels of mini dystrophin that's in the muscle biopsy samples. I will also say that we expected that based on the work that every one of these companies done with the dog. The dog model of Duchenne is very severe and very like humans. And each of these approaches were tested in the dog model. We tend to restrict use in the dog to this type of genetic approach. And in those tests, we've seen a remarkable reduction in the phenotype in the dog. There's videos that I'm sure many of you will have seen online where one of the dogs, even at 7 years of age, you know, is running around, looks quite normal. That's a dog called Jelly. And a dog will typically die within the first year. You know, so this mini dystrophin was expressed and was highly functional, and that's what we're expecting to see in humans. So the data that we've got right now is one where we've expressed dystrophin, mini dystrophin. These are in generally younger, healthier boys, so we can't say an awful lot about what the functional activity is. But going out over the next 2 to 3 years, there's where some of these questions really will be answered about how robust is the effect. You know, how does it change the phenotype of the boys? Does it make them stable? How much does the protein stay around for? Is there a need to redose? Does this stay around for a year? Is it 10 years? Is it 20 years? Like, one doesn't know. So there is a real ton of questions that have to come out. And what I'm trying to do is not to get too excited about the results right now, because I think that they're exactly where they should be. But there's a ton of information that's going to come out over the next 2, 3, 4 years that really will guide us about how robust this is and how much it's going to change. And I think that there's no doubts that what we're seeing right now, this is likely to be a very important part of therapy going forward. And it certainly could likely change the way that we think about treating this disease. So as far as I'm aware, the Nationwide have treated up to 4 boys right now. The exact same in Pfizer. I think they've treated 4. They've told us no information about that. We're not going to learn information from Pfizer about either the muscle biopsy samples or any other time function tests for at least another year. It'll be in the middle of next year that data's been due. And Solid have dosed 1 boy, and they're now, as far as my latest pickup in this is, they're now recruiting further boys in the site down in Florida, so that there's open for recruitment. So I think it's really exciting times for each of these, but there's a lot that we have to learn, and that information is not going to really be revealed until we go through the next year to next 2 years, and that there's really what parents want to see. You know, what is the robustness of this effect? Any questions there, by the way? Please. Yeah, I don't have a really solid answer. Like, I can say that and I don't know if it's 30%. Just I'm going for about. Oh, the question was about the 30% of boys that have significant levels of neutralizing antibodies, and what's one doing about that? I don't know if the number's 30%. You know, I'm taking a stab at that based on there's approximately 30 to 40% of the population have got very high titers to these. There's 2 different AAV serotypes that have been used, the 7.4 and AAV9, and the neutralizing antibodies that we've seen cross-react with both of those. There is ways to reduce neutralizing antibodies, and it's not straightforward. And what I think is going to happen over the next couple of years is we'll look at different ways in order to try and reduce it in those boys that where it is high and it's not going to be applicable for therapy. Because I can imagine as these drugs get approved, they will be fairly expensive, and insurance companies will probably want that as a prerequisite, that titers should be low. So I think that there's certainly scientific ways that we can do it right now. It isn't easy, it's not straightforward, but going forward I think there'll be a ton of work that goes into there. One of the things I showed today, Deborah mentioned on her slide earlier on, there's a company that we're working with called 4D Therapeutics, and I will tell you that it's the thing that keeps me up at night whenever I'm thinking about what's the weak links in the pipeline that we have. And I recognize I'm fairly bullish on what we're doing with these mini-gene approaches. I think that there's a really good chance that this will have a significant impact on the disease and will be approved. So the limitations here is how do you redose if we have to go back in after a couple of years? Or how do you catch those boys that have got very high levels of neutralizing antibodies? The basis behind what 4D Therapeutics have done is they've designed new viruses. So there's about 10 or 11 serotypes that we can use. They've designed hundreds of millions. And out of that they've found some that transduce muscle much, much more efficiently than either AAV9 or 7.4. And I mean hundreds of times more efficiently. And what that means is the dose comes down 100-fold. They've also identified ones that do not cross-react with the neutralizing antibodies that are out there for natural AAVs, and are they much less at cross-reacting with those. So I think the future work has really been led where if we expect this to happen, the next couple of years is going to be transformative with new AAV approaches into this. And that there's real, it's a long answer to your question, I'm sorry about it, but that there's really why, because it's important, we're thinking ahead and thinking that we will get into a position with these new AAVs that are out there that have never been seen before, that we've identified right now, could take over from some of those and actually give you a broader reach into the boys. Okay.
So are they finding boys with the lower tolerance for AAVs? What's the test?
So the question was, how do they find these boys that have got lower neutralizing antibodies? Each one of the clinical trials have got that as an inclusion criteria. So the boys will come in and they'll literally run, one of those antibody tests on them.
Is it a blood test?
It's a blood test. Yeah. It's pretty straightforward, but it's not routine. It's not commercial. You can't go out and get it done somewhere else. It's only in that clinical trial center. It's part of their inclusion criteria. Jim.
So just a functional question. So we've got these three choices of gene therapy studies, and theoretically, or actually in practice, families are screening for, or hoping to screen for multiple studies. If you screen fail for solids, is there any chance that the test for the antibody, because it's a slightly different, that you get into Sarepta, is it worth people trying?
Yes, it is. It is, and I've seen examples of that there where it's not that if you fail one, you might fail the other, but it's not. You don't have to get to a point, hey, where it's zero interaction. You just want it to be very low, and so absolutely yes, you need to go into each one of them. Any questions?
You said that Pfizer and Solid are also doing the same sort of thing. On the first one, there was the deletions that they've included in the clinical trials. On these two slides, there's ages, but there's not deletions.
That's
are
right.
their deletions the same? Like for those of us who have kids with very rare deletions who aren't included, would these two be open to that, or are they also going to have a closed range?
Yeah, so it's actually a very good question. No, for both, as far as I'm aware, both Solid and Pfizer have got no limitations for any specific deletions, so it's all comers into that. The one that they've done, the one that Jerry's done really is based on his experience on previous gene trials. So he wants to have certain things in there where you don't have epitopes in order to, build up antibodies to what they're trying to insert. So I imagine going forward they'll relax that, but they just want to give their trial the best chance of success at the very beginning. Great questions, guys.
Still, I have one question. Is there any company in US or in the rest of the world who are recruiting kids who are not on steroid for a gene therapy or something?
No, the question was, is there any company that we're aware of that's recruiting boys for gene trials that are not on steroids? And the answer is no. They may not want boys to stay on steroids all the way through, but part of the recruitment criteria into this, the inclusion criteria, is they'll want boys on steroids for at least a short period of time beforehand and during this, because they want to try and immunosuppress slightly, so that you don't get a big immune response to the virus that's going in.
Yeah, and one of the things that I'm having trouble is most of the companies have exclusion saying that there should not be any behavioral or cognitive impairment.
Mm-hmm.
And that is something where my son is having the issue with.
Yeah.
So that is another reason why.
It really depends on the trial. So the question was about having patients that don't have any behavioural or cognitive impairments. For some trials where the boys have to go under a significant amount of instructions in order to follow time function tests, that there's really usually the basis behind that. I don't think that's going to be the case in these ones.
So there is another company called Genethon. Do we have any update on them?
No, as far as I'm aware, so the question is what's going on in Genethon. Genethon's a French company based out of Paris. They've been involved in gene research for quite a while. They've forged a relationship with Sarepta, I believe. So Genethon are still preclinically, as far as I'm aware. I don't think they're actually in clinical development, but they're following a very similar path. I should also say, it reminds me, as I'm thinking to myself, these are mini dystrophin genes. There's a couple of companies that are involved in mini utrophin gene therapy, doing exactly the same thing. So they're packaging a mini utrophin gene into the AAV. They're still in preclinical phase. I suspect that if they continue to go down the pathway they're going and get funded, that there could be in the clinic in about a year or so's time. Now, the difference behind it, so that parents are aware, a number of these boys are dystrophin null. They've zero dystrophin. So if you put in, the fear may be that if one went in with a mini dystrophin into a boy whose body has never seen dystrophin, it might build an immune response to that dystrophin. It's seen as a foreign protein. But everybody's got utrophin. Even Becker boys or Duchenne boys have got utrophin in them. We all have it right now. And so the basis of the theory of actually switching to a mini utrophin gene is that you will not and should not build an immune response to that. So it's going to be interesting to see how this evolves. Now, the way I would describe it to you is there is marked enthusiasm, as you can tell the way that I'm talking about it, for this approach. But I think if it continues along this pathway, it will be an important part of therapy. You've got 3 different companies, and now you layer on to that to the fact that there's a couple of companies actually earlier than this actually thinking about utrophin and if there is a problem here. So there's a lot of bandwidth that's built into this that parents should walk away with thinking the next couple of years in this type of research is really going to be transformative in our thinking about how we treat boys and what you want to do with them.
There was a study by Summit on utrophin upregulation.
Yep.
It didn't work out. So why will this gene therapy of utrophin work?
That's a good question. So the question is, why would it work whenever Summit failed? They're very different approaches. The mini utrophin is actually the mini utrophin gene that's inside the virus, and you're going to infect muscle, and it'll actually transcribe a protein that you will make. The Summit.
But if everyone produces utrophin, why we need to put gene?
It's to do with the quantity. If you put it in genetically, then you'll be able to vastly increase the amount of mini utrophin compared to natural background utrophin levels, which are typically very low. Okay. The Summit compound failed for reasons that's nothing to do with the genetic approach. They had a small molecule approach that was trying to upregulate the amount of protein that's normally produced within muscles. And it failed not really because of the science or the utrophin pathway. It really failed because the molecule was very heavily metabolized. There wasn't enough of it, of the parent compound actually circulating around in order to test the pharmacology.
Do they publish this kind of detail in any of
I don't know.
molecule level detail, like why it didn't work?
They should do. I'll have a look and see if there's something out there. Yeah, the question was, did they publish any of that? But I'll certainly check and see.
So the reason I'm asking, because I'm considering anti-myostatin by Roche, but there was a failure by Pfizer, so should I go for it or not? So
Yeah, I think you should. We'll get onto that in a little bit. Yeah, we'll talk about that. Any more questions on the genetic approaches? Yeah.
Is there any consideration of a one-size-fits-all approach where
Yeah, that's going to be, so the question is there a one fix all or one fits all approach into this, depending upon the mutation? Like if you go in the CRISPR pathway, it's again going to be very personalized, highly personalized, really based on a mutation, because you're going to have something that's going to guide, you know, the sequence into fixing or finding something that's very unique to that individual. So it'd be highly personalized medicine. The only one that I could get close to saying is going to be general for everybody is these genetic approaches with the mini genes. You know, there again, that should be applicable to all comers, independent of any mutation that's out there. Oh, there's one other one. This GAL-GT2. This is not one that puts dystrophin back in. This is another protein that compensates for it. It's meant to upregulate utrophin. It's not as advanced as the other programs. It's still at an early stage of clinical development. I think there's only one patient that's been tested right now. But I put it up for just completeness. You know, it really sort of sets the stage where it's not all just mini dystrophin. There's another one that's out there. I don't need to say much about this. Everybody knows deflazacort's been approved. But much more interested in here is the novel anti-inflammatory approaches. You know, steroids and when to start steroids and what dose you should start on or what the frequency to start on is probably one of the earliest and toughest decisions that parents have to make. You know, and there's a number, you guys included, you know, that haven't started yet. And it really speaks to the need, you know, that parents have. You know, is there alternatives out there for me? And there's where this comes in. I think it's going to be an important part of the arsenal as you go forward, you know, and really think about how do we actually manage the disease, particularly with these new anti-inflammatory agents. And there's 3 companies that are involved in this. First one I've got up here is vamorolone from Revergen. This is a steroid derivative. It's a novel steroid derivative. And just to explain a little bit, steroids have got a fairly complex biology. They actually turn various genes on and genes off, and that there really explains the complexity of it. Some of those are good, some of them are bad, and that there gives rise to the myriad of side effects that you see with steroids. What these guys have done is really to tease apart the good and the bad. They've really focused on separating out that there are steroid effects on the anti-inflammatory pathways from the ones on bone or the ones that are on various other components that are associated with the side effects. This dissociation has been incredibly challenging. The industry have been trying to do this for a while, and they believe that they've got a molecule that does it. In the non-clinical setting, you can see that there's separation between the positive effects as an anti-inflammatory agents away from other properties, and they seem to have shown that in the clinical setting. Right now, the guys are in a phase 2B trial, recruiting 120 patients for what could be a registration trial. That's recruiting right now. The data that they've presented to this point seems to suggest that there's a separation, but it's too early to actually, at least for me as a scientist, to say, yes, I've seen all of that. The data's not complete, and that's the basis for this phase 2B trial. They'll run through a number of doses. It's placebo-controlled, and that there, by next year, we'll have a real complete picture of what this molecule is actually doing. But for your point of view, I think really recognizing for those who are not on steroids, and this could be a trial that individuals really want to look at and take a close look at the number of centers that are going on in the US. The second one that I have here is a company, again on the East Coast and here up in this area, Catabasis, and they've got a really innovative approach in order to try and mimic what a steroid is doing. There's this particular bit here, it's called NF-kappa B. That's a molecular pathway that steroids use to exert a very potent anti-inflammatory effect in this disease. And Catabasis have taken an approach to target NF-kappa B specifically without using a steroid molecule. So their product opportunity is one where it does not have a steroid structure, it doesn't have the steroid side effects and liabilities that come with it. And the focus that they have is really to target that particular mechanism with a compound that's got 2 properties. They're recruiting their phase 3 trial right now. I've put the details on this. You'll find that in clinicaltrials.gov. And the company's planning a fairly extensive recruitment campaign over the next year or more in order to confirm the findings that came out from their phase 2 trials. And the last one that I'll mention on this is a company called Mallinckrodt. Mallinckrodt have got an unusual approach into trying to either look for something that can replace or reduce the need for a steroid. And that's the focus in here. They've got something called a corticotropin analog that stimulates the release of natural cortisol in the body, as well as acting on anti-inflammatory receptors. And the basis behind this is it will either drive a steroid-like effect using something that's not a steroid, or reduce the amount of steroid that a patient actually needs. They're recruiting in a number of sites in the country right now, Florida, Atlanta, and the like, as well as Canada and European countries. Too early to say what this is. Like unlike the Catabasis product or unlike Reverigen, this isn't injectable, so it's quite a different way of actually administering. So you take a number of factors into consideration as you look at that. But a different pathway yet again. And the basis behind this is you have a number of companies looking at different ways to try and either reduce or replace steroids. And that's going to be an important component as we see this data play out over the next short while. Any questions on there? No? Okay. So, please.
Is doing anything on
Is-
GCSF?
Say again?
GCSF based approach?
Ooh, that's a good question. The question was for GCSF. I talked about that last night. So that it's a granulocyte colony stimulating factor, and there's some data out there in one clinical trial that shows that over the period of a year, that there was a fairly significant effect on time function tests in these boys. I don't have any slides about it, but I'll say a few words. The data was truly interesting. They injected the boys monthly for the first 3 months and then quarterly for the rest of the year. And there was a clear impact in the data as far as 6-minute walk test and strength measurements, which you might expect, because you might expect actually nascent muscle stem cells, to regenerate new muscle. It's a single trial. And so I think that it really merits someone going into a controlled trial. There's a couple of companies that sell that. Amgen and Thousand Oaks in California is one company that sells that their product that is approved. And so I personally feel that someone should take that and actually run a controlled trial so that we can actually understand it and look at real data out of that.
So as CureDuchenne, can we do something like to fund or something?
Let's talk after this. I'm happy to have a conversation about it. You can tell I was really interested in the data that came out of that, but I always have to be cautious. And it was an open label trial. There was everybody knew what they were on, and it always makes it a little bit more difficult to actually read the data. But it was interesting enough for someone to actually say one needs to do a control trial in there so that we can understand both any efficacy and any risks associated with using that. And the dose that they used, if I recall, it's a long while since I read the paper, but the dose they used was probably only about half the recommended therapeutic dose. So there's a number of questions that I would have before I'd say, yeah, that was real. Okay? Fibrosis, I mentioned this at the beginning. I think it's going to be an important part of how we look at and manage this disease. Fibrosis is really considered very much the end product of muscle degeneration, chronic inflammation in muscle. And being able to manage this here is something I think that's important going forward. There's one company, Fibrogen, that's really focused on this. They've got this antibody, it's a connective tissue growth factor antibody. That they've been using in phase 2 trials. The thing that I'll tell you, I met with the company no more than about 4 weeks back, really to get an update on what they were doing. And this here is a very specific mechanism that is actually involved in the fibrotic pathway. And they're interested in exploiting this in a couple of different indications. One of them is IPF, pulmonary fibrosis. And a number of months ago, they actually reported very positive primary endpoints were met with the drug with a very good safety profile in pulmonary fibrosis. They're obviously looking at it in Duchenne. Their Duchenne trial is fully enrolled. They're not recruiting any more patients. It's what's called the data's being locked down right now, and the database is being cleaned. But we will get top-line results out of that before the end of this year. Based on a couple of things. One is its target, which is connective tissue growth factor, which is involved in the fibrotic pathway, and the success and safety that they've seen in IPF. I'm curiously optimistic, that we'll see good data out of this, but we will see this data before the end of the year, and then the company will not only announce that, they'll make a decision on what their regulatory strategy will be. So that's something to watch as we go forward. Muscle regeneration, we touched on very slightly. We did lose Pfizer. That was a real surprise to a lot of people, particularly those in Pfizer that were involved in the early stages of this program. It's not clear yet why their program failed. It's clear that it failed, but it's not clear yet why it failed and whether it was a mechanistic thing for Duchenne. and you can't turn it up anymore, or whether it was another fault of their antibody. This was Pfizer's second attempt at actually taking a myostatin antibody into the clinic. This second one I thought was going to be much better than the first. But I've no reasons, I've no insights, I should say, on why that there failed. But I can tell you that the Roche compound actually is continuing to perform well. They are recruiting into a fairly significant trial. The data, they've presented a number of posters recently, about 4 weeks ago at the World Muscle Meeting, and again reinforcing the very positive nature of that, particularly in relevant models for Duchenne. So all I can say is I don't know why Pfizer failed, but it doesn't distract from my enthusiasm for this. And I'll add in that I think that's going to be an important component. Being able to generate muscle in these boys, that if you think about it in the natural history, whenever these boys are 7, 8, 9 years of age and you're on the declining phase, you're on the declining phase because you're not generating muscle as fast as you're losing it. And that becomes important in here. So I really want to see this here continue to be pushed and pursued, even though Pfizer stood back out of it. So we'll see what happens. The data that they've generated is positive so far in non-clinical models, and that's all they've published. They've reviewed their phase 2, and they're moving on to a much larger trial, which I think is a positive sign. We won't be able to read anything until this trial reads out, and that there's going to be next year. And I've got a collection here that I dropped into other approaches. Everybody's aware of what Synthera is doing. This is a product, again, for older boys. It's really for respiratory function and those declines. Their phase 2 trials showed positive result. The FDA has given them very clear guidance on what they need to do in order to get an approval, and they've set that up into a much larger phase 3 Citrus trial. That's recruiting right now. It's going to be a couple of years before we see that there data, but the trial is looking for patients, and that there's something that we'll watch and follow on very carefully. Capricor is one that we looked at quite early and were intrigued by a couple of things. As I talked about the Citrus trial and looking at older boys because of the respiratory issues, this here really was one that was focused on older boys, was focused exclusively on cardiac issues and particularly dilated cardiomyopathy. We've talked about this before to give you guidance, but their trial read out with some fairly positive results in the phase 2, and that was a single injection of these cardiac stem cells into the heart, and they followed the boys for over a period of a year. What they learned through that was they probably need multiple injections, and so they've set up this phase 2B trial. I'm trying to find my own reading on this. Oh, it's a phase 2 trial, 84 patients, non-ambulatory. What they're doing right now is not going straight into the heart. They're actually going IV with exosomes from those, and that we believe is going to actually give you a broad systemic effect beyond just the cardiac effect. They picked that up in the non-clinical model. So this is quite an interesting product opportunity that we again will follow closely. They're recruiting patients right now in a number of sites, and next year we'll get the data out of that for the first time and be able to understand it. Yeah.
Would that help boys that already have the cardiomyopathy and maybe strengthen the heart?
Yes, so I'll explain a little bit about what I jumped over it very quickly. The earlier trials that they'd done before they got into Duchenne were in older patients who had quite a severe heart attack, and they injected in these cardiac-derived stem cells, and there they were able to show increases in the strength of the muscle, increasing in pumping efficiency, and remodeling of fibrosis, so there was less fibrosis that was in there. And that was the basis for moving forward into a Duchenne setting. So they took older boys who had existing dilated cardiomyopathy, and that was the basis of the first trial. It was a single injection straight into the heart with a certain amount of stem cells, and at the end of the 6 months and 12-month period, they were able to report out and see some remodeling in there and some improvements in pumping efficiency, and that was the basis for what they've done right now. Now, let me explain something about it. They were using these cardiac-derived stem cells that don't engraft into the host. So I'm not a big stem cell fan. I'll get parents that will ask questions about it or want to go to Mexico or some other strange companies and get some of that done. I've never advised anybody to do it. The difference here is that these stem cells do not engraft into the host. And the question would be, what do they do? If they don't engraft? The effect is really something that's released called exosomes. That's the whole belief in the mechanism behind this. And they're packages of information that actually switch on and regenerate nascent stem cells that are in there. And whenever they've done this in the non-clinical model, they showed not only improvements in cardiac function, but because you get systemic exposure, you get it in skeletal muscle as well. And that's the basis behind this trial. What they're doing there is infusing these into the boys. They're going to get them, I believe, once every quarter, if I can remember it rightly. And they'll be looking at 6-minute walk tests as well as cardiac outputs and various other things.
So do the boys have to have like a certain percentage of like ejection fraction percentage in order to qualify?
Yes. Yeah, and that's predominantly why they went into older boys.
What would be the percentage?
Ask me after this and I'll look it up for you. Okay? I will do that. And I think the last one that I have in this category is really the Italian company, Italfarmaco. And they've repositioned a drug called gavinostat. Gaviноstat's something called a HDAC inhibitor, histone deacetylase. That's got a multitude of actions. You know, it's fairly far upstream, and I think it's just going to have some anti-inflammatory activities, muscle regeneration activities, things like this here. So it doesn't fit into a single little bin. They've done a European trial that was open label and got positive results out of that. That's really propelled what's going on right now with the US and European trial. This is a much larger trial that's recruiting right now, placebo controlled. So they're doing a fairly detailed trial in a significant number of sites, both in North America and in Europe. We won't get data out of this for a year and a bit, but it'll give you a sense of what's actually going on in the background as we start to get all of the information that comes in over the next short while. So I could have stood and talked for like twice that amount of time about the stuff that's going on preclinically, but it's great to focus and spend our time talking about things that are real, things that you should know about, because thinking about what's going on in clinical development right now is either looking for people to participate and ones to recruit into it, or the data is going to read out in a time frame that allows you to think about, this is what I'm thinking about for my son. The next 2 or 3 years, we'll have data on almost everything that I've spent the last little bit talking about. So we're going to be incredibly informed over the next short while. And I had a slide like this at the beginning that gives you a sense of what's going on, so it can remind you and you can sort of walk out of here going, yeah, I need to pay close attention to that. Or I need to get hold of that guy and talk to him and find out more about this because it's important to me. But you can see these RNA-based approaches, and the importance is really driven by what nature's taught us with Becker muscular dystrophy patients with shorter RNA. And you can see the exon skipping from Sarepta, Wave, NS Pharma, and a few others. PTC with the nonsense read-through. I've talked a bit about the CRISPR-Cas, and I really didn't want to say too much simply because it is preclinical. The work that's being done is really exciting. They're doing really solid dog work, and that data is reading out to show very high levels and really high levels of genetic correction. And that there's something I think will play its way into the clinic. The gene replacements we spent quite a bit of time on. Utrophin is not a dead program. There's another company out there that are in, I've put that in italics only because it is non-clinical, it's a preclinical program. The fact that Summit program failed was more of a reflection of their compound rather than the mechanism. We weren't able to test that mechanism in the clinic. So there's another company that's going to come up in the next short while that'll move into the clinic trying to modulate utrophin that I think is an important approach because it's one that is not going to be dependent upon an individual's mutation. If you can turn that protein up and keep it up, then it could actually alleviate the disease, and it could be something that could be quite important. The other ones that are in there, you can see how this is going to come together. I won't read all of those, but how this combination approach, where I started to talk about the next few years, is about having these read through and having choices of approved drugs that are going to work together. And the muscle regeneration is important for the reasons that I said. As boys start to decline, being able to regenerate that there. So that's it. How's my time, by the way? Okay, I'll take another couple of minutes just to sort of give you a sense of what's on the horizon, because I won't spend time on this. But like I said, whenever we chat a little bit, I do stay up at night, worrying about what we've got, and where we are, and where are the weak links in this. I think this is a really, really exciting time for Duchenne research. I said earlier on that technology has finally caught up to this disease, and you can see all of that technology in full play in these clinical development programs. I come from a world in biotech and pharma where the failure rates of clinics is really high. Attrition rates can run at about 80 or 90%. maybe not so much in rare diseases, but that's certainly quite typical whenever you look at why clinical programs fail. So I look at this as going, it's fantastic having all these programs and all these backup programs all targeting the same sort of thing because we think it's important, but not everything is going to work. It's not. That's just simply life in this business. And a lot of what we do is trying to recognize what else do we need to fill in and what else is important. And that's where some of these gaps are that out there. These are programs, for the most part, that we've worked with or we're really focused on and trying to move forward into the next stage. These new antifibrotic programs, there's one, the CRISPR-Cas9 programs I talked about, new exon skipping ones, new targeted variations of AAVs to try and get around some of the limitations that we see today that's going to change the landscape. And I think Deborah talked on that very briefly, monoclonal antibody-driven exon skipping drugs in case anything else fails or this turns out to be much more efficient. That's probably as much as I think I want to say, but I think you can get a sense that we don't think this job is done. And you shouldn't go out of here either thinking that. Like, it's taken a long time for all of these programs to get aligned. Like, I totally salute the companies that are involved in this because this is really tough work. We couldn't do it without the companies and we couldn't do it without the people in this room. And not just for financing, but being part of what goes on in clinical trials and participating in each of these. Like, it really takes a whole team effort to make this thing work. And we're thinking about what's next. And that there really is part of the struggle that we have to understand that there's no gaps in treatment or there's no gaps if programs fail and the next one's going to come on. So a really important time. These are programs that we funded over the last number of years with help from people in this room and across the country. And it's really helped to forge what the landscape looks like. I think if I'd have talked here whenever I first joined Deborah and the team about, you said 9 years ago, it doesn't feel like 9. But I'd have been here, and because I'm Irish, I probably would have talked for an hour anyway, so, but I could have done it in 5 minutes because there wasn't that much going on. Okay, but right now, like, I think being able to work our way through this and get used to understand, that there really is important because I want you to come out of here really thinking and being thoughtful about why all those programs are there and why they're really coming together.
So I'm really interested in master protocol and after speaking with other rare disease groups that are starting to look at that, when do you think Duchenne is going to be at a place where we can start, we have 2 approved drugs? I mean, I know we're not quite there yet, but when do you think and how are advocacy organizations and companies going to start sort of working together to
We have Lauren. Yeah, this is about at what point in the near future would one envisage a time whenever we have master protocols that actually cut down on the amount of time that's needed to open up clinical centers? Yeah, and so Lauren's actually, I'm going to have a cup of my tea here, but I'll let Lauren come to the microphone and actually say a word about it because it's really part of what TRINS is thinking about in the way that they're operating. And there's a number of other organizations that are imaginative like that there. It is important, but it really speaks to the complexity of the different approaches and different companies and different centers that we're using worldwide. Do you want to say a word about it?
So I think there's definitely a lot of efforts being made towards the master protocol, and I think models that you can kind of think about are the natural history studies, because those essentially create the kind of baseline that you want to keep adding possible interventions. I think there's models for which it worked, and there's a great publication from the New England Journal of Medicine with members of the FDA that were the lead authors on those to see and provide information on how that could be created. I think what becomes challenging with the master protocols is, as Mike just presented, is there's many different interventions. So how do you kind of put in the same bucket people that are eligible for certain exon skipping or mutation-specific interventions versus those that are not? And so I think it just becomes challenging to be able to find something that's going to work for everyone, where you can have blinded portions, non-blinding portions. Different companies are in different stages of their developments. So I think it's a great idea, and I think it can potentially maintain some of the resources that we have. I think we're a rare disease community. We want to maximize our resources, being able to, patients, exactly. And so I think that's very important, of finding ways we can maximize what we have and build on what we've existed. what we've created. So I think a lot of what we've done with TRINS is building on what the network has done for Synergy, using these platforms similar to natural histories that can be a baseline to then add patients into those.
Do you have any sense of a timeline or when we can start looking for, people like have their children on a therapy, both approved therapies, my boys are on it. It's the end of the road for us at this point. This is great research. Now we're waiting for another drug approval, but at what point do you think we'll start adding in those that are on therapies and starting to stack to improve upon the outcomes? We had great outcomes. How do we get better outcomes? Do you have any sense of a timeline? I know everybody's been talking about this for years, but in terms of who's gonna take the plunge and start that?
And I don't have an answer to that one. I think nobody really does. I think Deborah introduced it when she welcomed everybody here, and I think Mike Kelly probably is somebody who supports this, is it's going to be a combination of different interventions. And so at what point can you start adding additional interventions based on where those are in their development? It's tricky, and probably more preclinical work has to be done on these combination therapies, because sometimes you just don't know how they're going to interact. Do you have a better answer?
Nothing else, I guess, everybody in the room.
Yeah.
Any more questions? Sure.
I got a question from my friend who is also the father of patient kids. He said that even though you start with a steroid, there is a study that out of 100, 10% of the kids do not get a functional benefit from the steroid. Is there any reason behind it?
Yeah, so the question really is, it's a steroid question, and it concerns out of a certain number of boys that will start steroids, there's a small percentage of them that will not get a functional benefit. I got to say that answering questions about steroids is always incredibly difficult, because not everybody starts on the appropriate dose. Not everybody is on the same steroid. Not everybody's on the same dosing frequency. Some are on alternate days, some are on weekends, some are on every day. Some are on low doses, some are trying to get close to the recommended therapeutic dose. And as the boy gets older and heavier, they don't want to adjust the dose, because there's side effects. And it's tough to answer a steroid question. And it's simply for that reason. There's a trial that's been going on for the last couple of years that really is set out to try and get to the bottom of that, where they're comparing prednisone with flazacort, with weekend dosing, with appropriate dosing, and trying to understand both the differences between them and the differences in the frequencies.
What is the trial?
I think is that the MOVE DMD trial?
No, 4 DMD.
4 DMD.
And they are trying to understand why it is not helping some kids.
Yeah, what they're trying to understand is a comparison between steroids and the dosing frequency and the dose. You want to say a word?
Right, so I think they're supposed to have study results in the summer. So it's been a trial that took many years. They enrolled a very large number of patients. It was actually funded in part by the National Institutes of Health. And so it's a combination with Newcastle University and Rochester University in the US. And so that should be, they do have a website. It's 4DMD, I think, .com. Or maybe .org, just because of the funding. But I've been told they're supposed to have readouts of that study in middle of next year.
Oh, thank you.
Okay, you can have some lunch. You obviously listened and asked questions. Bon appetit.
Thank you.
You're welcome, guys.